My name is Vula Saradakis. I'm the head curator in the Collections and Archives Department at the Griffin Museum of Science and Industry, as well as a NASA Solar System ambassador. So on behalf of Griffin MSI, I'm delighted to welcome you all to our 13th annual From Science Fiction to Science Fact event. And this is our look inside the amazing ideas that are being developed to explore the solar system and beyond in partnership with the NASA Innovative Advanced Concepts Program, or NIAC for short. Now, if you haven't already had the chance to visit the museum's newly renovated Henry Crown Space Center, which opened last May, we encourage you to stop on by, and the updated exhibit features exciting new additions, and the space has been completely redesigned, highlighting how everyone can play a role in space exploration. So today's event is bound to spark questions, and during the program, you're welcome to type in questions into the Q&A box that should be at the bottom of your screen. And at the end of the program, our guests will answer as many questions as they can in the time that's allotted. So let's get started. I'm proud to turn things over to a longtime friend of Griffin MSI, Kathy Riley. Kathy is the Strategic Partnerships Manager for NIAC, and she's going to share more information about the program and introduce the NIAC fellows you'll be hearing from today. So turning things over to you, Kathy.
Okay, thank you very much, Ula. We're very happy to be here today and excited to share a lot of cool technologies with you. I think we have a video to show first before I get started. Can we? So I'm going to share my screen in just a second, but I wanted to mention that the video shows that we push boundaries, that we dare to defy what might be possible. And we've got over 300 studies from very talented people who might be changing the future of space exploration. So with that, I'm going to go ahead and share. So tonight's talk is about science fiction to science fact. What is in the realm of science fiction might actually be technically credible and become science fact. I will be speaking along with Frank Spellman from the NIAC program, who will be running Q&A, as well as NIAC researchers Robert Hinshaw and Lynn Rothschild from NASA Ames Research Center. I'm Kathy. I'm the Strategic Partnerships Manager at NIAC, and I've been running this program with MSI for 13 years. And the Q&A is a heavy lift, so Frank is very engaging and a wonderful speaker, so he will be handling the back end and all the questions everybody might have. So thank you, Frank, for being here. So what is NIAC? NIAC is the NASA Innovative Advanced Concepts Program. It helps support early studies of innovative visionary concepts that could one day change the possible in aerospace. So what does that mean? It means that we develop early stage concepts for NASA missions beyond the next generation, which is 20 plus years out. We are extremely popular with the public. We're open to ideas that are not quite ready for prime time, but each one is technically credible and highly vetted. And the most exciting thing is there are many spin-off technologies that develop from these early stage studies that help humankind on Earth. Our studies are very high risk with the potential for high reward. We're advancing technologies, we're not just a propulsion shop, or we pretty much advance technologies across all scientific disciplines. So in order for exploration to take place successfully, you need technologies to take them there. NIAC, going to give you a few little examples of early NIAC studies that actually turned into missions. We inspired the Ingenuity helicopter, also known as Ginny, which is the first aircraft to achieve power control flight to another planet. Also, an early study that developed from the Jet Propulsion Laboratory, it was Mars Cube 1, also known as MARCO. It was the first deep space CubeSat mission. CubeSats were not used for space until this mission proved that it was successful. It was a real-time communications data relay with technology demonstration, and it greatly reduced mission costs. So it opened up sort of a new realm. It was revolutionary and opened up a new realm of space exploration. But we also do NIAC technologies that are for space initially, but actually transition to Earth. We have optical mining by Transastra, which captures and removes space debris. How does that relate to Earth? Well, removing space debris could potentially save our satellites or other communications. And we also have contour crafting, which was 3D printing using regolith, which is the dirt, and the silica, which is sort of the sandy, glassy type of surface of the moon, and how that could be melted and sintered into habitats or landing pads. And that transitioned to Icon, which is, I think, a multi-billion-dollar company now that is advancing 3D construction here on Earth. Icon's a very interesting company. You may want to look them up. And then we had something called Superball Bot, which was a tensegrity robot where the payload was in the center and it was sort of squishy on the outside. So you could drop it from great heights. And that has transitioned to firefighters in the Houston area who are using it for sending it into hazardous environments for chemical, gas leaks, wildfire detection, and even for military uses. So why are we here today? We're here to discuss, can we use life as a tool? And by life, I mean the microbial world. So the first to speak is going to be Robert Hinshaw. I'd like to introduce Rob. He joined the Radiation Biophysics Laboratory at NASA Ames Research Center in 2023. And he was a NASA postdoctoral program fellow designing automated biotechnology systems for space exploration. Prior to that, he earned his PhD at MIT in 2022, and he was developing neural cell culture models to study the interaction between radiation exposure and Alzheimer's disease. So he currently leads the NIAC-funded MitoMars, which he'll talk about. It's a project that's investigating the potential for mitochondrial replacement therapy to heal cellular damage caused by space radiation. So we'll learn about that. And I'm going to introduce both of our fellows, our researchers, and they'll just speak one after the other. Lynn Rothschild is an evolutionary biologist. She's an astrobiologist and synthetic biologist at NASA Ames Research Center. Lynn's also an adjunct professor at both Brown University and Stanford University. Her research explores how life, especially microbes, evolved on Earth, and she uses those lessons to understand the potential for life beyond our planet. She founded NASA's program in synthetic biology, and she explores how can it play a pivotal role in the future of space travel. Her interest in synthetic cells is twofold. Number one, how can it inform the origin, evolution, and search for life elsewhere? And number two, how they could aid in space exploration and settlement. She's been named a NIAC Phase 1 Fellow 5 times, so we call her the Queen, which is a testament to her groundbreaking work at the intersection of space exploration and synthetic biology. The NIAC program is the most open-ended and far-reaching of NASA's technology programs, and we are always open to new ideas. So look us up and maybe even apply. So I'm going to stop sharing here and we're going to pass it over to Rob.
All right. Thank you very much, Kathy.
Thanks.
All right. If you cannot see my presentation, just interrupt me and we'll go back to troubleshooting. But thank you all for taking some time to come listen to us talk about our projects. I'm thrilled to have the opportunity to tell you a little bit about our crazy idea, and I hope that by the end you think maybe it's a little bit less crazy and a little more credible. Yeah, so in short, we are aiming to replace astronauts' mitochondria to boost their deep space endurance. As Kathy said, I'm Rob Hinshaw. I'm a postdoc fellow here at the NASA Center out in California, just a little bit south of San Francisco. And my co-investigator on this project, Colin Headley, is at Stanford University next door, and the VA here in Palo Alto. So I'm going to break this talk into three quick parts. First, I'll tell you a little bit about radiation exposure in space, which is the motivating problem for doing all this in the first place. Then a very quick biology lesson about mitochondria, how they function, why we think they're important, and then I'll bring them together and tell you about mitomars. All right, so in NASA's best guess or plan for sending people to Mars, we'll have astronauts off the Earth for about 2.5 years. This is laid out in something called the Design Reference Mission 5, if you want to look it up. But essentially, it's a 6-month transit in interplanetary space from planet to planet, then 500 days, a year and a half or so, on the surface of Mars while the planets revolve and come back into position for the 6-month return trip home. There's a lot to do to get ready for 2.5 years of complete independence from Earth. Not the least of those problems is the radiation exposure. There's a lot of radiation in space, of kinds that we don't see here on the ground, which one makes them hard to study, but they're also, in addition to being hard to study, very much more damaging than a lot of the species we see on Earth. And this won't mean anything to you yet, but we estimate that astronauts on this trip will accumulate about 1 sievert of radiation exposure. I'm going to put that into context for you for reference. That unit sievert usually has a milli in front of it when we talk about human exposures. The career dose limit that NASA has set for its astronauts is 0.6 sieverts. So you can already see the problem here. You can't abort the NASA mission or the Mars mission 60% of the way through. Once you're heading away from Earth, you're locked into that mission. So we need to either find a way to reduce the amount of radiation those astronauts see, or lessen the impact that radiation has on their biology. And we're taking the second tack. So before I get into that, just a little intuition for you guys on how this space radiation interacts with your biology. There are many different kinds of it, but what makes it particularly different from what we encounter here on Earth is how densely ionizing it is. So if we take something like gamma radiation, which is also in space, and you also find it on Earth, we call it sparsely ionizing radiation. It's essentially it's light. It's very high energy light. In this image, that pale green oval in the background is the nucleus of a cell with all of its DNA. And those bright dots are clusters of DNA repair proteins that have congregated to fix the DNA after the radiation damage. And your cells can do this pretty well. Unless you get too much too fast, your cells can repair themselves and life continues. In space, however, we have this other kind of radiation. We call it it's a specific type of particle radiation. We call them HCEs. They're very highly energetic, highly massive particles. These things are kicked out of exploding stars near the speed of light with all their electrons ripped off. So they're growing very fast. They have a lot of mass. They have a lot of charge. They're really good at damaging matter as they wreck through it. So this is another cell nucleus, the blue thing in the bottom. And those bright green dots are still DNA repair proteins, but you can see they've clustered in coherent tracks. These are, that's the footprint of an individual radiation particle, 3 individual radiation particles traveling through that cell and leaving behind a very dense field of damage that is very difficult for the cell to repair. And if you run the numbers, it turns out that in this 2.5-year trip to Mars, we think that those astronauts will have to contend with about 7 of those really heavy particle traversals in every cell nucleus of their body over the course of that mission, which is not great news for them. Although we think we can do something about that. Before I get there, a little bit about why we think we have a handle on this. This little bean-shaped guy is a mitochondria. you might know it from your grade school years, is the powerhouse of the cell, which indeed it is. But we also think that it might be the primary radiosensitive target of the cell when it comes to space radiation exposures. All you need to know about him right now is that he's got two membranes, an outer membrane and an inner membrane. And that inner membrane has a bunch of good stuff on it. You don't need to know any of this. We're going to put a box around it and just call it the electron transport chain. Its job is to push positive charge in that space between the membranes and negative charge into that inner space. And then this other guy, ATP synthase, uses that gradient to generate a molecule called ATP, which is the energy currency of the cell. That is what all of your cellular biological processes use to function. Without that, nothing works. And that's the main purpose of your mitochondria, is to generate those guys. The problem that your mitochondria has to contend with is that process is very oxygen hungry. That's why we have to breathe so much of it to live. And when you put those free negative charges and oxygen together, they create this chemical species, or a whole suite of them, that we just call reactive oxygen species. But they like to crawl around and chew on things that we don't want them to chew on, and they break stuff, like the electron transport chain and ATP synthase. And it gets a little bit worse than that. Mitochondria also keep little packets of DNA in that inner space. It's like a quick reference manual for repairing proteins in that electron transport chain. So yeah, fun fact, not all of your DNA is in the nucleus. Some of it's in the mitochondria. And those reactive oxygen species also will come chew on that DNA, mutate it, render it less than functional, which is not great for your mitochondria, but they've been doing this for a while and achieve a pretty steady balance. They've got several processes to repair the DNA, check it against other copies, prevent the damage from happening. And under normal conditions, you sit at a happy medium. There's an acceptable amount of error that your mitochondria can take from this chemistry, and they chug along. The problem is that when you take these things into space, which in the history of life has not really been done before, these space radiation species, particularly these highly damaging particle species, are very good at making these reactive oxygen species. When they hit biology, most of what they hit is water, and they break the water molecules apart. The pieces recombine in weird structures that are very chemically reactive and can go break things. And we think that this is a key mediator of spaceflight induced damage. So what that looks like in the context of your mitochondria, this happy balance between production of ROS and energy and damage and repair, you get a radiation particle ripping through it, and suddenly you swamp the system with a ton of extra reactive oxygen species. And then all of the natural processes that keep your mitochondria happy and healthy can't keep up. Things mutate, things get damaged. Your mitochondria can't produce enough energy. Your cells start to shut down. They leak damaging chemical species out to the rest of the cell, to the rest of the tissue. And all of that equates to effectively what we think of as like an accelerated aging phenotype, which is a suite of terrible things that you would not like to have happen to you, not the least of which is an elevated risk of cancer. There's been a lot of work over the last couple decades put into preventing this from happening. Like, how do we prevent the radiation from hitting the astronaut in the first place? How do we sop up these reactive oxygen species, make them less damaging, help the mitochondria shift that balance back to good? But what my co-investigator and I thought about was, maybe what if we just swapped the mitochondria out? What if we just got rid of the damaged ones and gave them fresh ones and let them start over? I was shocked when he told me that he had a way to do this, but he does, he can. So what we're going to try to do is transplant mitochondria to repair radiation damage. And here's how that looks. First, starting with the astronaut, before they go on their mission, they're on the ground. All we need is some blood. We culture those blood cells and expand them, isolate mitochondria from those blood cells, and then bank them, put them in a box, essentially, in stasis. We can shield that tiny volume of stored mitochondria and send it along with the astronaut on the mission to Mars or wherever you'd like to go, and then deliver those shielded, undamaged mitochondria back to the astronaut over the course of the mission, hopefully reducing the impact that radiation has on their biology. Which is all well and good, but a couple pieces of magic have to happen for this to work. First, we need to be able to protect the mitochondria from the radiation. I don't have, unfortunately, too much time to talk about that today. What I'll talk about instead is the other half of the equation, which is when you give the mitochondria back, they have to get to where they need to go, and they have to function like normal mitochondria. So to do that, my co-investigator has developed a really ingenious way of addressing these mitochondria to different parts of the body. We call them mitotags, but they are antibody conjugated mitochondria. So an antibody is a protein with a variable shape at the end, so you can change its shape to essentially tag it like a lock and key to any part of the body. So we can make these mitochondria specific to heart cells or a kind of kidney cell or endothelial cells, or just send them and have them taken up widely across the body. So those little antibodies will attach to a receptor on the outside of a cell that will tell the cell to engulf it and then incorporate that mitochondria into its native network. And then it can afford to recycle the damaged mitochondria. And this works in practice. What you're seeing here is a real image, a fluorescent image of cells. The blue in the center are the nuclei. This looks like one that recently divided. And the white stringy stuff is all of those cells, native mitochondria. They don't look like little beans because in practice they are all networked together in this filamentous structure. And then the red are our antibody tagged mitochondria that have integrated into that cell's mitochondrial network. And we've known that this is possible for a long time, like we scientists in general, but the problem has been that the cells don't really like to take up foreign mitochondria. So on the left here is another image. The colors mean all the same things, but the untagged mitochondria, which are red in this image, there's not a ton of them. But if you add the antibody tag, you get the image on the right where you get like very good adoption of those external mitochondria into the cellular network. And we've done some functional tests on these guys, and we can see that the cells recover from reactive chemical damage much faster when they have these fresh mitochondrial boosters. And we've even, or Coco has essentially refreshed the immune cells of old mice with fresh mitochondria and shown that they can fight off infection better, which is pretty exciting. So we know that not only do they get to where they need to go, but they actually work when they get there. What's left to do is demonstrate that we can store these outside the body, preserve them for spaceflight, and that they work in the context of radiation. And then the really big picture is that we can create a library of these tags for the mitochondria that address them to any body system you like. And then we can potentially have a handle on any condition that involves mitochondrial dysfunction, which is almost everything that you associate with aging. So we've talked here about spaceflight's an obvious use case, but so are things like neurodegenerative diseases and obviously mitochondrial diseases, skeletal system problems, reproductive system problems, your immune system, your guts, cancer, your heart, really anything that wears down over time due to mitochondrial dysfunction, we think that we can boost it. So that's it in a nutshell. I'm so fortunate to have an incredible team working with me on this project, and we're really excited to see where this goes. So we'll do questions after Lynn gives her talk. That's it in a nutshell, and I'll stop and hand it over to Lynn. Thank you all very much.
Rob, that was incredibly cool. I was really excited to see what you were going to present.
Thank you.
Okay. I give a lot of talks and I feel like everyone starts with and now for something completely different. So and now for something completely different. I'm going to zoom out a bit in a more big picture way and then zoom in a little bit more to the project that I'm going to present today. So imagine that you're on planet Earth. You don't have any difficulty talking to someone who's sitting next to you or picking up the phone and talking to someone even on the other side of the planet. But as we go forth, we have all sorts of things that we now have that are our current issues, but we have this problem of a distance and a time break. So for example, humans on planet Earth have all sorts of needs. We need transportation, we need life support, we need oxygen, we need medicine, we need clothing, and so on. We need habitats to live in, and as Rob mentioned, there's a lot more radiation out in space, and so the habitats and the protection become even more important. We need power, heat, and light because we don't have that infrastructure the way we do on the Earth. Again, we need the radiation protection. We need forms of communication, and just because this gentleman's waving a wad of cash, we need money as well to get there. So as we go off of planet Earth, we as humans still take these needs with us, but now we have other sorts of issues, so what I call challenges for humans beyond Earth. And one of the big ones is getting these materials up there. So you order something from Amazon and it comes in a plane and a truck or a train or whatever. It's not really a big deal. But when you launch something into space, you're fighting Earth's gravity well. If you've ever watched a launch, a lot of the spacecraft is jettisoned in the first few minutes. And so therefore, you may not worry if you have a piece of equipment in your lab or a television set or a computer or whatever, and it weighs a couple of pounds, or it's even hundreds of pounds, and it takes up the whole table. It doesn't really matter to you when you're at home, but when you have to launch against Earth's gravity, well, every one of those pounds makes a big difference. And so suddenly you want everything miniaturized as much as possible. So this up mass and up volume is really important and tied into our cost issues. You also need things that are able to be stored, that are reliable, and they're flexible. You can't just get to the moon or Mars and say, oops, I forgot something. You have to be able to make do with what you have. I think of this as the ultimate camping trip. Again, you need the power, and I started thinking of this really as the ultimate circular economy. I know on Earth we talk a lot about recycling and making sure that our trash is recycled into new metal and glass products and so on. But when we're in space, the recycling becomes even more extreme. So now every water molecule becomes important and every carbon atom becomes important. So that's why I now have started calling this radical self-sustainability. And so we need to do all these things, and the humans have all these needs that we're taking with us. And NASA's been really good at using the technology that has an on-off switch. But I think that there's another technology that really can be much more effective for some of NASA's and other space agencies' problems. And just to cut to the chase for you, that technology is life itself. If you really think about it, life is like this micro machine that does carbon chemistry and nanotechnology with incredible precision, much more than any scientist with all the degrees and the honors and so on will ever be able to do. So, starting with this idea that life is this technology that can solve a lot of problems, we've been exploring some what we call big ideas in my lab over the last 15, 20 years or so. And some of them are things like the fact that life does chemistry on the Earth. And I don't know if you can see in the background, there's a cup of sake and, various other fermented products. This sort of course, if you look at a chemistry catalog, a lot of these molecules are already made by life. Even aspirin originally came from willow. So why not do that off the planet? When we make DNA now, we don't physically sit there and have to use another organism. We can print DNA, the material, the genetic code for life. But what's really cool is that we send the information to make a sequence of DNA to a company that does it. And then they send back this physical piece of DNA. And this may not sound like a big deal, but it is, because for the first time in the entire history of life, we've now broken that physical continuity. So if you're going to send information to make DNA that then goes into a cell across town or across the planet, why not to another planet or beyond? So now we're able to break this. We also rely on nanotechnology on the Earth. And when you hear nanotechnology, you start thinking about big clean rooms and all sorts of infrastructure. But it turns out that life is an incredible nanotechnologist. So we've explored things, for example, like using the DNA double helix, not as a genetic piece of information, but actually as a building material. And it turns out you can do really cool things, including making wires out of it. But no time for that today. But on the subject of electrical wires, we generate electricity. So I don't know how many kids are in the audience, but if you're an adult, you ate dinner tonight or lunch or breakfast in order to fight entropy so that you didn't break down. And part of that energy has gone into making electrical currents. That's what's allowing your brain to work and your nervous system to work. The electric eel just happens to be good at releasing it all at once. So perhaps we'll someday be able to use life to convert chemical energy to electrical energy off planet. Now, what about drugs? It turns out that most of the pharmaceuticals that we use only are tested for about 3 years shelf life. And as Rob pointed out, we're talking about a round trip to Mars of about 2.5 years. And worse than that, many of the new drugs that are based on proteins are not stable without refrigeration. And even with refrigeration, they're only stable for about 6 months. So again, no, it doesn't work for a 2.5 year round trip. We can do things like biomine. We can use life as a way to extract metals. Again, love to talk about that, but not today. We can also do things to alter life so that they can live in more extreme environments, or maybe create life fresh, what I call life 1.5 or life 2.0. And so now maybe you can give this characteristics that we didn't have otherwise. In fact, we've got a NIAC phase 1 project, which is finishing up, using life to detoxify the perchlorates, these chemicals on Mars that prevent us from drinking the water or using that water for rocket fuel. So many of these projects, as I say, we've been doing in my lab, and big shout out to NIAC because many of these have been funded by NIAC, my absolutely favorite program at NASA headquarters. That's no secret. But so, we've got life that does all these incredible things, and you start thinking about life, well, particularly the materials and the chemistry. We're now starting to talk about things like sheep and earth, silkworms and cotton plants and rubber trees and so on. We're not going to take sheep or silkworms or rubber trees with us. But what we might do is take these tiny organisms. This is a bacterium called Bacillus subtilis. And if you look carefully, you can see many of them have little white dots. These are spores that they form that enable them to live out in the soil for long periods of time. And in fact, it's so good that these spores also are able to live outside of a satellite. This has been proven for nearly 6 years. So why not use genetic engineering and take these capabilities of making rubber or wool or silk or cotton or whatever, and now engineer something like a Bacillus subtilis to make those, and then we can take nature's hardware store with us. So what have we done about this? And that's really the main thing that I want to talk about today is this NIAC program that we've had that's now in phase 3, and that's looking at habitats off planet. And so here you see the Apollo astronauts, and if you're the first or the second or the 50th or the 100th person to go to the moon, by golly, that's got to be the most exciting thing in your life. And so fine, if you have to sleep in a little lunar module for a few days, fine, because this is so exciting, you can deal with it. But as we're starting to look to have astronauts living on the surface of the moon and then ultimately on Mars for long periods of time, you can't ask them to be functional for long periods living in very tiny, cramped quarters. And so the idea is you take a brig habitat with you, something that could potentially house several astronauts for months or maybe years on end. And the sort of thing that's been discussed by NASA is having a spacecraft or some kind of habitat that's about 29,000 kilograms. So a lot of metal, you can count on it. We call it, it's like a turtle. A turtle has its habitat on its back, its home on its back all the time, but then it's got to lug it around and it's tired. So maybe we can do something better. We can be more like a bird and use the materials that we find to make our own nest or our habitats off planet. So we do think there's a better way. One suggestion has been to have a robotic construction of houses out of the surface material, what we call the regolith, or maybe out of some of the water that's there and build ice houses. And there are problems with these approaches. And so we thought, I wonder if there is a better way. And so, oddly enough, we turned to something that you're all familiar with, and that is a mushroom. Well, not exactly a mushroom. Mushrooms are the fruiting bodies of many fungi. So this is what's produced for reproduction. That's actually what you're eating. But it's created out of a dense mycelial network that actually is even more visible underground. So this is the real body of that mushroom. And you can see in that gray circle, that's a picture I took through the microscope of this mycelial network with these tiny hair-like hyphae in it. So that's really the main body of the mushroom. And if you look at it, it's all in the soil. So out in nature, these mycelia will bind soil particles. And so other people have realized that if they can bind soil particles and certainly bind wood, because many of them live off decaying wood, why not use this as a way to build materials? And so there are a couple of companies now that are based on this kind of technology, but we thought, maybe this would be a way to grow a whole habitat off planet. And this is one of a couple of incredible pictures I'm going to show you by my main partner in crime on this, Chris Maurer, who's the principal architect at Red House Studios in Cleveland. So this is how we envision a habitat on the surface of Mars. I'm not going to show you the whole process today that we use to envision this, but basically the mycelia are sent up in a dried form, perhaps with dehydrated wood chips, but they're in an inflatable. We're not just going to put them on the surface of Mars, and then they wouldn't know where to grow anyway. There's no stable liquid water, same thing on the moon. So what we do is put them in an inflatable, and that's kind of a fancy word for a balloon. Or the way actually I think about it is like an inflatable jumpy thing that kids use in the summer, or an inflatable raft or something like that. But now instead of just having air in it, you've got fungi and maybe dehydrated wood chips or some other kind of nutrient there, so that the fungi can now start to grow and fill this inflatable, and you have a habitat. And as you might imagine, if you're taking this inflatable with you that is packed up just like you buy a balloon and it's all packed up, or a raft, or any of these things that I mentioned, and you have very small amounts of dried fungi and some maybe dried material, but you take the water that is found on the moon or Mars and add it and grow it on site, you've got all sorts of advantages that other approaches don't have. Particularly that you now have a really low mass approach. You don't have to worry about hauling all this 29,000 kilograms of steel into space. It's also inexpensive, and it's a great material. It's got lots of great psychological properties. It's going to remind you of home. It's going to have more of a woody, natural feel than stainless steel. And also, think about it, when you build a house, you often have construction crews coming in and clearing out a flat area. Well, we don't have construction crews on the moon or Mars to clear out a nice flat area. That would just add more up mass. And so here you've got a system that could grow to fill some of these craters and so on. You don't need to worry so much about that kind of things. And at the end of its life, you could in principle compost it and grow your potato like in The Martian. And so we started calling this green architecture for the red planet. So as Kathy kindly mentioned, we had a phase 1 on this a couple of years ago, looking at applications for Mars. And in the phase 1, we looked to see, is there a there? Is this really totally crazy, or is this got a potential for working? And we decided that this definitely did have a potential. We went into a phase 2 where we started studying this in greater detail. And one of the great honors of my life, we got a phase 3 last year. And so we've now really started to focus very hard on transitioning this to a really functioning system. So one of the things the program pushes us on is to have a customer. Now, unlike a company, it doesn't have to be a paying customer, but we identified 2 potential customers, people or programs that would be actually interested in having us develop this technology. Of course, an obvious one is NASA or other space agencies to build these habitats on the moon or Mars. And so we're working towards that. But in the meantime, we've identified another one, and that is the commercial space stations that are starting to come up. And particularly, we've been in consultation with Starlab. So let me explain that Starlab is one of these commercial space stations that got seed money from NASA to get going. It's a consortium of a couple of companies. And it's a little bit different than something like the International Space Station in that the modules are going to be launched as a single station. So it doesn't have this big construction in space sort of thing that the International Space Station has done. And that means that we can outfit it on the Earth and just have it launch all at once. Now, if you're reading the fine print there, you might be surprised to see Hilton hotels. It turns out that they're doing the living quarters. So what we've done is look both at the laboratory working space, and here's one of their mock-ups, and we think that you can do better than just all stainless steel, that we could have some mycotecture panels in there so that we would feel like it's a more sort of natural, potentially woody type feel. And also, this particular material that we're making has a lot of great properties in terms of acoustical dampening. So the hearing's going to be better, and vibration, and we think it is very flame resistant, but we're in the process of doing those tests right now. But what we think would be really cool is to push our luck and try to get into the living quarters too. So recently, at the invitation of the vice president of Hilton Hotels, we were able to pitch some of this. And so I put together this little slide of the Hilton hotel room that I stayed in Washington the night before we visited him. And this was actually their favorite picture in my entire presentation. I took a picture of the room, and you can see all those little mushroom icons. These are all the things in a hotel room that I believe that you could replace with a fungal mycelial approach, all the way from hard structures like the floors and the walls. And you can see my bag over on the left, the back of the chair, or even the whole chair, the tables, and so on, but also some of the soft materials. So, for example, the bedspread or the curtains, because there are people working now on making fungal products that are more like imitation leather. And so obviously we want stuff to be cool. To me, the biggest compliment we get in our work is when someone says it's cool. And so I'm going to show you a quick video that Chris has put together for his vision of how a living quarters should look. Now, remember, today astronauts sleep by being strapped to the wall. And if you're in microgravity, okay, you're strapped to the wall. But it's in the future, again, we're going to want something a little fancier, but you don't want to float away. So I'm playing the video right now, and hopefully you can see this. This is Chris's vision, which I think is very cool. So you have a bed-like structure. You can see the astronaut on the left is starting to float away, but instead he's got an air compression pushing him down onto a sheet. Same thing with the one on the right. And there they are in bed with sort of a terrestrial feel, but yet keeping them from flying away. Now, you may notice also that these mushroom materials are black, and that's because we've discovered over the course of the last year that the pigment, the coloring in the black fungi will help prevent radiation damage. So maybe it will help Rob out as well. And so it also, kind of looks sexy having these black mycomaterials. But in fact, it's really there for the radiation protection as well. You spend a lot of time in bed, so why not? And I'll just give you one little quick bonus slide. As part of our attempt to push forward into the future, we've also come to the realization that astronauts don't do laundry in space. And so we did a quick calculation, and it turns out if you have 6 astronauts going round trip to Mars for 2.5 years, and they change their clothes as often as astronauts do on the International Space Station, which may not be as often as you do yours at home, you would end up with 2 metric tons of laundry. And as I tell everyone, I'm a biologist, but even I could design a washing machine for under 2 metric tons. Fortunately, we have colleagues in the United Kingdom who actually have taken a lot of really advanced technologies, including using very little water and acoustic focusing and all sorts of things, to develop ways to wash clothes that are very gentle, including dry cleaning. And so we've developed a system that I've been encouraged by NIAC, although it was not funded by NIAC. It was funded by another program in the early stage innovation at NASA, and has just been picked up by the Mars Campaign Office. So in addition to all the fungal materials, it looks like we're going to be developing a way to wash those and other things off planet. And so I'll just leave you with this picture of our vision of a fungal habitat on the surface of the moon. So I think back to you, Kathy.
Yes, that's right. So we're going to get Lynn, you stay on camera. Robert, if you could, and I'd like to introduce Frank to everybody who will be running the Q&A, and we'll get started. I think there are some questions in the Q&A section.
There are. Thank you, Kathy. Thank you, Rob. Thank you, Lynn. And of course, Jason and Bula for having us. My name is Frank Spellman. I'm the program analyst for NIAC. I'm really honored to be here, not only with my colleagues, but all the guests tonight. So thank you for coming. I'm going to get started with some of the questions in the chat. Oh, by the way, if you can use the Q&A function, that's part of Zoom to submit questions. If you're shy or remember a question later, you can always email us at hq-nyac@mail.nasa.gov. So let's get started here. This is a high-level question for Lynn. Can you define mycotecture?
I'm really
Briefly.
glad you asked. It's a portmanteau. Okay, so it's taking two words and jamming them together. Mycology is the study of fungi. So that's the myco, it's the fungi. And texture is the last part of the word architecture. So it's fungal architecture. And it's a word we invented. So that's why you haven't heard of it before.
Perfect. Also, just a note that the recording link will be shared by email for everybody in case you missed something or had to step away for a moment. Okay, Rob, this question's for you, and I think this sort of gets at the distinction between active shielding, passive shielding, and what makes your mitochondrial replacement therapy unique. So if you find a material to protect the mitochondria, then can't you make a spacesuit out of the same material?
Yeah, great question. So the fundamental problem with shielding is that the best way to shield from radiation is to put mass between you and the radiation. And mass is the very thing that is heavy. That's what makes heavy heavy. So it's expensive to launch. So if you're going to shield an entire spacecraft, or even just like the sleeping quarters of the spacecraft, it takes a lot more mass, a lot of lead, a lot of water, what have you, different types of radiation, like different materials. But if you can take the mitochondria out, store them in, say, a small volume this big, then you can afford to pack a bunch of shielding around it, which is just infeasible to do for a large structure. So it's just a matter of scale. But yes, ideally we would love to shield our astronauts with a ton of radiation protection. It's just it's not within the realm of what we can do yet. And hopefully, when we get to the surface of a planet, we can build some mycotecture domes and have that put more mass between us and interplanetary space.
So if you're in one of our mycotecture beds, that'll help you.
Yes, exactly.
Exactly. That's why I was glad you brought it up, Lynn, in your presentation. Speaking of other things you brought up, Lynn, can you and would it be useful to bring electric eels into space, into Mars?
Probably not a great idea. I think that would be taking away from other mission capabilities. But once again, if you study the actual cell biology operation of converting chemical energy to electrical energy, I believe that we can make a biomimetic. And actually, I've had various students over the years with electrical engineering degrees that have worked on little bits of that. And so I don't think that we want to take the electric eel, but we want to use that as an inspiration.
Well said. So speaking of things we want to bring, if we made the stuff out of mushrooms, and Lynn, this is for you, of course. If we made stuff out of mushrooms that were edible, could they eat them if they needed to? And that being the astronauts that were using these mycelial
Well, you certainly can
structures in space.
eat fungal mycelium. Yeah, the mushrooms are actually compacted mycelium. You certainly could eat that if you wanted to. We actually don't want them to form mushrooms, but I've even mentioned that you could build a table that has a long, skinny part in the middle that just allowed the mushrooms to grow. So you'd have a combination table and dinner coming out of the middle of it. So yeah, I think that you could throw that in as well, and that would be another very cool use of fungi. In fact, let me add that I was on a podcast recording with in Norway yesterday with someone from the for the Kennedy Space Center who is looking at the potential for fungi for food in space. So I know that people at Kennedy are looking at that specifically.
Excellent. This is a question that's not so much related to structures or mycelium, but to biology nonetheless. What about women who have a cycle? How did women astronauts and women who colonize Mars deal with that in space?
I'm pretty sure they bring their own
I
products
don't know.
with them. Yeah, I'm pretty sure they bring their own
Yep.
products with them and they just have them there.
Of
A
course, they
relevant problem
haven't been to
nonetheless.
Mars yet. I suppose mycelial tampons. We'll have to get back to you on that, obviously.
Q&A inspiring maybe a next NIAC proposal. All right, this is a very real and practical question here, Rob. When do you foresee getting to human trials if you plan on doing that, and are you subject to FDA approval?
Yes, we would be. So anything medical that goes into a human, at least that's produced in this country, needs FDA approval. And yeah, we very much hope to be doing clinical trials in the near future. So we first prove that it works safely in animals, and then we get to go ahead to do clinical trials. That process can be very long, but if there is a need, it can be accelerated. And if we think that this is likely to go faster than most drugs because it's not a chemical drug, it's a biologic. So it's a mitochondria. They're already in your cells. So we expect to have an easier ride through that process. But yeah, we're into predicting the future now, which is a little tougher to do. But hopefully it's something that we can measure on years and not tens of years.
Excellent. That helped answer a couple of questions in chat, actually. Back to you, Lynn. What's so special about mushrooms that you would want to build a civilization on another planet with them?
Well, it's not that my heart's in mushrooms. I really love algae, if you really want to know. But it's that these fungal mycelia can do all sorts of things building these materials. So I alluded to the fact that there are companies working on fungal leathers. And as you know, Hermes, the high-end company, has now does have a line of fungal mycelial bags, and much to my surprise, has invited me to their headquarters in September. So producing things that are sort of similar to leather is one thing you can do. Here's something else that we've made. This is a brick. I don't know if you can hear it bouncing, but this is a compressed fungal wood composite. Smells not like a normal wood glue composite. It smells vaguely mushroomy, but you can see it's a very hard substance. This one was created by another colleague. It's more like styrofoam. In fact, it was designed as a drop-in for styrofoam. And so it's got great insulation capabilities. And so taking the fungal mycelia, there are all sorts of ways that you can tune the mechanical properties depending upon which ones you grow and how you grow it, and whether you bake it in the end or freeze it or just let it dry out. And then think about it, the mycelia will grow to fill any shape you give it. And so when you're building with, say, wood, you have to worry about the grain of the wood. You have to worry about the fact that trees only grow so big and they grow straight. And so if you want to go around a corner, you're now going to have seams and so on, and that's going to create structural problems. But with the fungal mycelia, we could actually just grow something to any shape you wanted. And that's why you've got these incredible bed shapes that I showed in the video. You can just let your imagination really go wild. And in fact, you can do other things with it. And I'll just plug one of our other NIACs over the years was a start of a program that's still ongoing, using the fungal mycelium as a way to bind metals so that you can use it to pull metals out of wastewater streams or use it actually for mining or for even separation of metals. And again, very easy to grow. These things will literally grow on garbage. Cheap, easy, can grow into any conformation you want. Get the black ones, get the white ones. You're good to go. So that's why I'm very bullish right now on using fungi for life's problems in space.
Excellent answer. Okay, Rob, what volume of mitochondria would be needed for a manned Mars mission?
That's a great question. And the short answer is we don't know. And that is part of what we are trying to get rough bounds on with this phase 1 NIAC. So stay tuned.
Now, this is a question for Kathy. Will you four be doing any other of these talks or anything like this in the future?
We do want a year here at the Museum of Science and Industry, but if you want more, let MSI know. We will be very happy to come back. We can bring the same fellows, but you might want to learn about other studies that we're working on that are crossing that line between science fiction and science fact. Thanks for the question.
Excellent. Okay. Rob, back to you. How intensive would the process be to get the mitochondria out of storage and ready to give to astronauts? Would they need a full dedicated laboratory on Mars to process them there?
Yeah, great question. So we hope that we can build this in such a way that you don't need any dedicated lab on the surface of Mars. The goal is to package it up as much as we can on Earth and have it as autonomous as possible for the crew. Getting mitochondria out of cells is a pretty easy thing to do in labs on Earth, and the idea is that we'll bank enough of them here before the trip so that you don't have to do any extraction and banking on the trip. Recovery from storage is that's the other big part of the puzzle. Cryopreserving or preserving in any fashion mitochondria is even a relatively new thing to do on the ground. So we're playing around with it to see what we need, what we can do, what we can't do. Hopefully, it's as simple as adding water. We think that we can get away with lyophilizing them, freeze drying them, essentially. But yeah, again, one of the major questions that we're investigating with this phase 1 NIAC. So you guys are on point with the questions.
Excellent. We have a question from one of our younger participants that says, I'm 10 years old. We have to watch out for meltdowns on Earth because of atomic radiation, and it's more powerful than other radiation on Earth. How would we prevent decay from atomic particles in that case? Would mitochondrial replacement theory be useful in that sense?
Yeah, absolutely. We think that it could be very helpful, not just for astronauts receiving space radiation exposure, but for people who get exposed due to any kind of radiation accident on the ground, or even people receiving radiation therapy for cancer treatment. You could, in theory, so one of the limitations of radiation cancer therapy is that you have to make sure the cancer gets the radiation, but all the healthy tissue around the cancer doesn't. And what you can do now with this, or what we hope we'll be able to do with this, is target those mitochondria specifically to the healthy tissues around the cancer and not to the cancer, so that you can give more radiation to the cancer and protect the healthy tissue around it. So short answer, yes, we hope that this will be widely impactful on Earth too.
Awesome. Great question.
The 10-year-old, he might want to be on Rob's team.
Yeah. All right. I got one for Lynn here. If the fungal materials are made here on Earth, can the spores be transferred to the Martian atmosphere or the surface and potentially grow in Mars soil? If so, how would that work?
Oh boy, let me sort of disentangle that. We could take spores, we could take dried mycelium. We probably don't want to take something that can form a mushroom I mean, we would for something that we eat, but not for something we want to grow a habitat, because can you imagine like a balloon full of mushrooms? That would, be a terrible material. You want the mycelium. So you can't really just grow it on the surface, as I mentioned. In fact, you can't grow anything because liquid water is not stable on the moon or Mars. And what I mean by liquid water is like you would drink out of a cup, in contrast to ice or steam. And so you already have to pressurize it and put it inside something to add the pressure. But then the last part of the question, I think, was using Martian or lunar soil. Unfortunately, we don't have soil on the moon or Mars. You say, wait, there's stuff on the surface. I can see stuff behind Frank. That's not soil. Soil's got all sorts of things like organic materials in it that allow plants to grow. What we have on the surface of the moon and Mars is something we call regolith. So it's more like ground rocks. And so things like fungi or even plants can't grow directly in the regolith at this point. We're going to have to treat it in some way. And I'm feeling awkward about saying much more because I submitted a proposal to you guys this morning on that very question.
Well, I didn't hear anything. All right. So related to that,
I didn't think you did.
Related to that, there's a couple questions in here about planetary protection. And so how do you contain the spores when you send them to Mars? Can you talk about that a little bit?
No, that's a really great question. And for those of you who don't know what planetary protection is protecting other places that might have life on it from life on Earth, and vice versa, protecting us from potential other life forms. Now, we don't worry about the moon because it's dead and we believe always has been dead. But Mars is still an open question And so now I'm wearing my astrobiology hat for this. And so we have to be extremely careful. There are treaties, the Outer Space Treaty, that says that we can't just go and contaminate the surface of Mars. But as I mentioned before, we can't grow things on the surface anyway. So our plan is always to grow things inside pressurized habitats. We're assuming that the astronauts are going to go through airlocks to go outside because they can't step outside either. I mean, the stable water means that their cells would just boil over. You would die very quickly, well before the radiation even would get to you. And so we do need to be very careful in terms of contamination, but I think our particular technologies are going to be particularly good in that area because we do have them growing inside inflatables. But I think your audience should be aware that as careful as we possibly are, I think that once we have humans on the surface of Mars, that human life is going to take precedence. So if it does come to a situation where an astronaut has to run back into the habitat and the airlock doesn't completely close right, we will prioritize the life of the astronaut over the astrobiology concerns. But that's why I think that we need to do as much exploration before we get humans there. And certainly we're required, obligated, both legally and scientifically, to be as careful as we possibly can.
Excellent answer. Okay, this is the last question, and Rob, this is for you. Could the mitochondria be used that you collect from one human be used on another human, or do you have to collect that same human's mitochondria and give it to the same person?
Yeah, it's a great question. We think that they're pretty interchangeable. So there is some genetic variation in mitochondria from people to people. They're called haplotypes. But even within an individual person, people have what we call a heteroplasmy. So they have different mitochondrial DNA throughout their body. So we think that largely we're going to be pretty robust to allogenic transplants. But to stay safe, we're starting with everybody gets their own mitochondria until we can prove that it's safe with animal studies.
Excellent. Thanks, Rob.
Okay, so thank you, everybody. Those were amazing questions. And that ends this year's From Science Fiction to Science Fact event. I just want to say that on behalf of everyone here at Griffin MSI, a sincere thank you to both Lynn and Rob for sharing their expertise and really exciting ideas with us, and to Kathy and Frank for working with us to help organize tonight's event. And thanks to all of you who watched our event this evening. We truly appreciate your taking the time to be with us, and we hope to see you again next year. Goodbye.